All right, well, hello everyone. So we're going to look here, and in this part, in the next several parts, what we're going to be looking at discussing is some basics of cardiac physiology. So we're going to be looking at essentially how our heart generates an electrical potential, an electrical signal, how it sends it, and then just some general overall functioning of the heart. So first thing that we want to look at is we want to look at the heart tissue itself, right? So we want to look at the cardiac muscle tissue, and that's what we see here in this, in the background of this kind of title slide here. You can see this tissue back here. This is the cardiac muscle tissue. This is the myocardium of our muscle. So when we look at our cardiac muscle tissue, it shares some similarities with the two other types of muscle tissue, right? So if you recall, we have cardiac muscle tissue, we have skeletal muscle tissue, and we have smooth muscle tissue, right? So what makes cardiac muscle tissue unique is some of its structure and some of its other physiological properties that we'll discuss. So one, the cells are going to be short and branched. So when we look at cardiac tissue, and I'll show you a picture here in a few slides, instead of being long and fibrous like we see with skeletal muscle, instead of being kind of tapered and spindle-shaped like we see with smooth muscle, cardiac muscle tissue is going to be kind of cylindrical but then branched as well, right? It has a sarcolemma, it has a plasma membrane just like skeletal muscle, as we've discussed previously. There are the T tubules or the transverse tubules. There's a sarcoplasmic reticulum with calcium. It's arranged into sarcomeres, so you have the overlapping arrangement of actin and myosin. Again, a lot of the same features that we see with skeletal muscle. At certain points along our cardiac muscle tissue, and I'll point it out here in just a moment, we have these structures called intercalated discs, right? Intercalated discs. What these intercalated discs are is they are essentially these junctions where one muscle cell meets another muscle cell. So where one cardiac myocyte meets another cardiac myocyte. And as a part of this intercalated disc, or these discs, we have two main things that make up these discs. One are called desmosomes, and one are called gap junctions, right? So desmosomes are a physical attachment. Okay, so physically attaching one cell to the adjacent cell, right? So they're mechanically joining two cells, right? The second type of protein that you have as a part of this intercalated disc is what's called a gap junction, right? Now you may recall when discussing cells and cellular junctions and the way cells interact with nearby cells, a gap junction is an opening, right? So hence the name gap, right? There is a protein that spans the membrane of both cell types, or both adjacent cells, both cardiac muscle cells, but there's an opening in the middle, so they forms this gap. And what it does is it allows the electrical impulse, as we'll see, which is just the flow of ions, just like we had with our action potential, right? It's essentially the same thing. Those ions are going to be able to move very quickly from one cell to the next, right? So what this leads to, in essence, is it makes the cardiac muscle cell and tissue, and makes the heart as a whole act as a functional syncytium, right? So this term here, functional syncytium, meaning essentially the heart functions as a single unit, right? So you get this very organized, very controlled and synchronized contraction of the heart, right? So if we look at some of the pictures of our cardiac muscle tissue, this is what we're looking at, right? So here we see our intercalated disc, again, where you'd have one muscle cell joining a nearby cell. We see our sarcolemma, or our plasma membrane. We have the transverse tubules, just like we had in skeletal muscle, right? You have your various organelles, nuclei, mitochondria. And then here you can see some of those other features, right? So you can see the desmosomes, again, physical connections between two cells, and then you have your gap junctions, right? And again, those gap junctions are going to allow the ions to move between one cell to the next, right? Now, again, you can also kind of see in here that overlapping striated appearance, right? So again, cardiac muscle is striated like skeletal muscle, but it's involuntary like smooth muscle, right? So again, it shares some characteristics and properties of both of the other cell types, right? So we can see another image here, something similar, but one thing we can see here is a section of the actual tissue, right? So a histological view, right? And you can see on that histological view, the image on the right, you can see the actual intercalated discs, and they're very unique and characteristic, again, of cardiac muscle tissue, and that's a good landmark. That's a good point to observe that tells you you're looking at cardiac muscle tissue, right? If you see those dark lines, again, you can kind of see some of them right here. You can see some of them down here. If you see those dark lines, that tells you more than likely that you're looking at cardiac muscle tissue, right? And again, in those intercalated discs, you have the desmosomes and you have the gap junctions as well, right? So with our heart, so if we know that we've got these if we know that we have these intercalated discs, and as a part of it we have the gap junction, we can then begin to discuss how you actually move the electrical impulse from one region of the cell to another, right? And so this is called the cardiac conduction system, right? The conduction system of the heart. And so we'll discuss it here in the next few slides. But essentially what we're doing is we're going from what's called a pacemaker, right? A pacemaker region of the heart that's going to initiate the impulse or the action potential, and then it's going to send that impulse across the atria down into the ventricles and cause the ventricles to contract, right? Now we have two different types of cells that we're going to look at, right? Two different types of cells that we'll see. We'll have contractile cells, and then we'll have these conducting types of cells, right? And again, we'll discuss both of them in time, right? So if we look at this pathway here, these terms here in bold, these are various landmarks throughout the heart that the electrical impulse is going to travel through, right? So first off is something called the sinoatrial node, right? Sinoatrial node, SA node. This is the pacemaker, right? This is the pacemaker. This is where the electrical impulse is initiated in the heart. So every time your heart contracts, where that initial stimulus for the heart to contract, where it comes from, it's coming from this region called the sinoatrial node, right? It's in the kind of posterior upper wall of the right atrium, right? So if I go back to this slide here, this image right here, that little region, that's your SA node, right? So we have our SA node. We have something called the AV node. AV means atrioventricular, so that tells you it's between the atrium and the ventricle. It's near the right AV valve, right, or that tricuspid valve. From the AV node, you move down into what's called the AV bundle. Sometimes it's called the bundle of His, okay, so you pronounce it His, like a snake, His. And this is running essentially in the interventricular septum, right, so the wall that divides the left and the right ventricles, this bundle of His is running kind of in the upper part of that wall. And then that bundle splits into what are called bundle branches, left and right bundle branches, or AV bundles, these bundle branches. And then from there, you then move into these last components called Purkinje fibers. And these Purkinje fibers are very wide in diameter. They conduct the signal very, very quickly. And these are emerging from the apex of the heart up toward the base, right? So they're starting kind of down at the point of the heart in the ventricles, and then they're moving up toward the top of the ventricles toward the base, right? And this is carrying the signal through the ventricular myocardium to excite the ventricles to contract, right? So you can see some of the components of this conduction system here, right? So we're starting in the SA node, the pacemaker. There are what are called internodal pathways, and these internodal pathways spread across the right and left atria, so to send the signal to the right and left atria. The signals then converge back to the AV node, and something important happens at the AV node, all right? Something extremely important, and the signal is delayed, right? So at the AV node, the electrical impulse, the electrical signal is delayed, okay? That's important, right? And we'll discuss why that's important here in just a little bit, right? But big thing, electrical signal is delayed at the AV node, right? After it's delayed and it continues, it moves down to the AV bundle, right, or this bundle of His. then splits into left and right bundle branches. And then from there, it moves into these Purkinje fibers, again, starting at the apex of the heart and then moving up through the ventricular myocardium, right? Just a little tidbit of information. The left bundle branch is a little bit larger because the left ventricle is larger, right? And you should know why the left ventricle is larger. It's larger because it's pumping blood against greater resistance, and it has to pump blood further, right, than your right ventricle, which only has to send it to the lungs, right? So that left bundle branch that leads to the Purkinje fibers going to the left ventricle, that left bundle branch is typically larger than the right, right? So for our control of our heart, right? So the innervation of the heart, the heart exhibits something called automaticity. Okay, the heart itself exhibits automaticity, meaning it has this own inert ability to stimulate itself, right, and generate an impulse to contract, right? So it doesn't need innervation. It doesn't need the nervous system, right? Now, that doesn't mean that it doesn't receive input from the nervous system. It does. Okay, it just doesn't need it. All right, but in our living bodies, right, in us as individuals, we receive innervation from our nervous system. We receive innervation to the heart. Right, our heart receives innervation. And where we get this from is we get it from our medulla, right? So we get it from part of our brainstem, right? So remember, you have the midbrain, you have the pons, you have the medulla oblongata, right? And in this medulla, so again, in the brainstem, you've got these two regions, one called cardioacceleratory and one called cardioinhibitory. And you can probably imagine what each of these different centers are doing with respect to the heart rate, so the number of beats per minute, but also the force of the heart's contraction as well, right? So notice here, this kind of last main bullet point here is that this innervation of the heart from our nervous system, and again, this is a part of our autonomic nervous system, right? So sympathetic versus parasympathetic. It is modifying, not initiating the electrical activity, right? So again, where does the initiation come from? The initiation comes from the SA node, but the modification comes from the nervous system, right? And so we'll look at that, right? So with the parasympathetic division, think back to nervous system, think back to the autonomic nervous system discussions you've had in previous courses. With parasympathetic innervation, right, that's your quote-unquote rest and digest, right? So conserving energy, right? And so in this case, what's that going to mean for the heart rate? It's going to mean that the heart rate is going to slow down, right? You're going to have a decreased heart rate, decreased force of contraction. The primary cranial nerve, or the primary nerve that's going to innervate our heart for this parasympathetic innervation is going to be cranial nerve 10, right, which is our vagus nerve, right? Remember, three cranial nerves carried preganglionic parasympathetic fibers. That was cranial nerves 3, 7, 9, and 10. So in this case, 10 is our vagus nerve, right? So some of these going to the SA node, some of these going to the AV node, and we'll look here in a little bit. We'll look at exactly how it works to slow down the heart. So here we're saying parasympathetic innervation decreases the heart, sympathetic innervation increases the heart rate. Here in a little bit, we'll look at how it does that, right? So how does it slow the heart when the cranial nerve 10 innervates it? How does it increase the heart rate when you get innervation from the cardioacceleratory center, right? So for the cardioacceleratory center, this is sympathetic innervation, increase the heart rate, increase the force of contraction, increase the amount of blood leaving the heart, which we'll call ejection fraction, right? Or stroke volume. So in other words, how much blood is leaving the heart with each contraction. May end up with an increase in blood pressure, things like that, right? So here we can see this kind of dual innervation of the heart, again with sympathetic increasing the heart rate, increasing the force of contraction, parasympathetic doing the exact opposite, right? So if we look at this left image here, again, what we'll see here is parasympathetic. These are these orange fibers, again, from the brainstem. And if you look here, which one is circled, it's our vagus nerve, right? So one going to SA node, right, primarily the right one going to AV node, primarily the left one, right? And then you have your sympathetic innervation. You have your cardio acceleratory center, right, sending signals down to regulate heart rate, force of contraction, things like that. And these are going to be, again, via some sympathetic nerves, right? So some cardiac sympathetic fibers going to the actual heart itself, right? So in this case, it's going to be these blue ones here, right? Or these are going to be the sensory limb, the sensory component, and then over here with green, this is going to be the motor component, right? So coming from, again, our sympathetic chain, again, a review, autonomic nervous system innervation, right? And the autonomic nervous system pathways if you need to, right? So this green one, remember, you always have a preganglionic and a postganglionic, right? So here's our ganglion, right, in our sympathetic chain, synapse with the postganglionic, and then here's our longer postganglionic to SA node to various parts of the heart for innervation. And then the right image here is showing the same thing, it's just not labeled. So again, here with our blue fibers, this is going to be parasympathetic, right? We're starting in the brainstem. So this, again, this would be which cranial nerve? This would be cranial nerve 10. And then here we can see some of our sympathetic cardiac nerves, again, merging from our thoracic region of our spinal cord, merging out to our sympathetic chain, synapsing, and then continuing down to the heart, right? So big thing here, like we talked about whenever we did autonomic nervous system, is think about what's the overall effect on heart rate, blood pressure, the force of the contraction. What's the effect of sympathetic versus parasympathetic on those different types of activities, right? So consider some of these questions, and then we'll pick up in our next part, and we'll begin talking about some more detailed topics related to the initiation of electrical impulses in the heart.